This Week in Microbiology is brought to you by the American Society for Microbiology at microbeworld.org/TWiM. This Week in Microbiology, episode number 3, recorded March 18th, 2011. Hi, everybody. I'm Vincent Racaniello, and this is TWiM, the podcast that explores unseen life on Earth. Joining me today from Princeton, New Jersey, is Cliff Mintz.
Hello, Vincent.
Good to have you back, Cliff. How are you?
Good to be here.
Very good. Today we have 2 new hosts on TWiM. The first one from Yale University, professor in the Department of Molecular, Cellular, and Developmental Biology, Joe Handelsman. Welcome, Joe.
Thanks, Vincent.
Good to have you on the show. Glad you could make it.
Great to be here.
And congratulations on winning the Presidential Award for Science Mentoring.
Well, thank you.
That's great. Do you get to go to the White House?
I did, and I met President Obama in the Oval Office, and it was absolutely thrilling.
Wow, that's amazing. I'm amazed that I'm talking to you. Great. Terrific. Our next new host from University of Louisville, the professor of biology, Ronald Atlas. Welcome, Ronald.
Hi, Vince. How are you?
Good to have you on the show. Glad you could make it as well.
I'm glad to be here with you.
And I hope that you and Joe could make some future episodes as well, because as you'll see after this one, you'll have a lot of fun.
Looking forward to it.
Before we go on to our 2 stories today, I wanted to just have Joe and Ronald tell us a little bit about themselves, where you were educated and what you're interested in. Let's start with Joe.
I did a PhD in molecular biology at the University of Wisconsin-Madison, and I studied symbioses between bacteria and plants, first rhizobium, and then I later moved into other symbiosis systems. I was a professor at the University of Wisconsin for 25 years, and then about a year ago I moved to Yale. My lab now works on symbioses between plants and microbes and caterpillars and microbes, and I love to talk about caterpillars, by the way. And we also do metagenomics on soil, trying to understand the metabolome of the soil.
Great. Perfect subjects for TWiM, of course. So how do you like the Northeast after being so long in Wisconsin?
Oh, I love the Northeast. I'm having just a fantastic time. Yeah, I grew up in the Northeast, so it feels like coming home, even though I only lived here for a few years and then spent 30 in Madison. But it really does feel like home, and I love being near the ocean and near the big cities. So it's ideal.
We stole one of your faculty just about a year ago. It may have coincided with you arriving, Shankar Ghosh.
Oh, I heard about him, but I never met him for exactly that reason.
He's now our chairman here in microbiology and immunology.
Ah.
But that's the way it goes.
Wisconsin's better than Columbia anyway.
Wisconsin or Yale? Which one did you mean?
Wisconsin. I worked at both places. I like Wisconsin.
So did
better.
you, Cliff, did you know Joe at Wisconsin?
Absolutely.
Yeah.
She was a couple of years more junior to me, but we've known each other for quite some time.
Very good. Well, that's great. How about you, Ron? Where are you from?
I'm also a product of the Northeast, although I've been in Louisville since 1973. But I did my undergraduate at the State University of New York at Stony Brook and then moved to Rutgers in New Jersey for my graduate work. When I showed up at Rutgers, I wanted to be an infectious disease virologist. And at that time, Selman Waksman took me aside and said, "Don't do that. We've conquered all those infectious diseases. We have 2 antibiotics. What more could we need?" Anyway, I was influenced by that and wound up going into environmental microbiology and discovering bioremediation and working on oil spills. So I still do work now for Exxon 20 years after the Exxon Valdez spill and for BP on their spill in the Gulf. But I guess I was into my career at Louisville for 10 years when I realized Waksman was wrong and all these infectious diseases were there. And so we started developing PCR methodologies for detecting pathogens in the environment, and that led me to being able to detect biothreat agents and getting into a lot of policy work, and like Joe, going to the White House on occasion, although my work was more in biothreat reduction work and biodefense. So I currently chair the Biodefense Committee for the American Society for Microbiology. I was president of the society when the anthrax attacks occurred, and that certainly kept me busy for a while.
So did you work on the anthrax investigation at all?
I did not actually work on the investigation, although I served on the FBI's microbial forensics task force, which was advising the FBI on developing robust methodologies that could hold up in a court case that went well beyond what we normally do in clinical microbiology or in public health investigation.
Well, it's just so happens that our first story, which I picked before I knew you were joining us, happens to do with the Bacillus anthracis investigation, which I guess they call Amerithrax. And a paper just came out in PNAS, I think last week, February 2nd. Bacillus anthracis comparative genome analysis in support of the Amerithrax investigation. And so I thought we would go over this, and it's great to have you here because you can give us the inside information. There are quite a few documents that go along with this paper. There is actually, the FBI has issued a very long 96-page document basically summarizing all aspects of the case. So as by way of background, I'll just mention a few things, and then we can start a general discussion. So I think everyone remembers that back in September, October 2001, envelopes containing spores of Bacillus anthracis were mailed to a number of individuals in the U.S., a number of senators, some media people, and also a place in Florida, Boca Raton. And they had spores in them, and 22 people got anthrax as a consequence, 11 inhalational and 11 cutaneous through the skin. 5 of the inhalational people died, and 31 people tested positive for exposure. A total of 10,000 people got prophylaxis because they were believed to be at risk for exposure. So that began a 7-year investigation by the FBI, which I believe they just concluded this past February, and they decided that a scientist at U.S. AMRID, Bruce Ivins, was responsible for mailing these envelopes. And I'm looking for the U.S. AMRID, United States Army Medical Research Institute of Infectious Diseases. Now, Ron, what happened? The FBI had developed a case, and then Congress asked the National Research Council to look at the science that was involved in that?
That's correct. There was a question, and there remains a question on the part of some congressmen that, in fact, Ivins was responsible. And so the National Research Council was asked to evaluate all of the evidence, and they, in fact, issued a lengthy National Research Council report recently, which again raises some questions about just how well science can, in fact, identify a specific individual.
So was this, the PNAS paper, which is basically a sequence analysis, was that a part of the FBI investigation, or was this separate from that?
It absolutely was a critical aspect of the FBI investigation. What came out of that investigation from the science side was the claim that there were a mixture of a couple of genotypes in the letters that were mailed to the Congress and to the press, and that, in fact, those genotypes matched the spores in a flask that Bruce Ivins had charge of, so that it was the mixture using whole genome analyses that they used as a fundamental aspect of the investigation.
So my understanding is, and correct me if I'm wrong, so the parent of the Bacillus anthracis of which spores were in the envelopes was the Ames strain. And when they sequenced that genome and compared it with the genome of the total, if you took out the spores and grew them up and sequenced that genome, there was no difference between the two. Is that right?
Yes.
It's only when you looked at the variants that were in the spores that you found some differences?
Well, when they plated it, they got several different morphological colony types, and it was the differences in those colony types that led to them pointing to the particular flask at Usamrid.
Right.
But you are correct that the fundamental initial analysis said that the AIM strain is the AIM strain. In fact, Bacillus anthracis doesn't exhibit a great deal of genetic variation from generation to generation.
And yeah, they mentioned that in this paper. And I think part of the reason is because it exists as a spore for long periods. Is that right?
I think that's true, and I think that's fundamentally different than the E. coli or many other strains that keep evolving quite rapidly. In the case of Bacillus anthracis, you get 99.99%, I believe, similarity no matter what strain you have.
Right. So this was amazing to me. The genome is over 5 million base pairs, 5,237,519. And then there are 2 plasmids, apparently, in this, and they were absolutely identical. I find that amazing to be able to say with confidence out of millions and millions of bases that they're identical. And so I think that is a testimony to the technology.
But also, Vincent, when you think about it, it makes sense for a spore former to not have that much genetic variation, because the idea is from an evolution, the way I was looking at it was kind of cool that if you're gonna die and then expect your progeny to exist through long periods, then you want to make sure they're equipped, if you will, to be able to do what they need to do to survive, and the spores need to contain and that information, and so it's really, really conservatively it's conserved, and it needs to be in order, because there's no real, if you're in the spore state, you're kind of quiescent, and there's no variation. So I think it's kind of cool that this happened, and the fact that using whole genome sequencing, like you say, with the abilities to find such small reproducible differences and feel very certain that they are the only differences is kind of amazing. But the thing that really struck me from this paper is the fact that despite the very, very small changes in one region, in a gene that is largely responsible for regulating sporulation in anthracis, that the phenotypes of the colonies were so different.
Right.
You know what I mean? It was like you were actually looking for the needle in the haystack, and they found it, and actually explained on a genotype level for accounting for what was responsible for the difference. But when you look at those colonies, they look very different. Very different, except for the last one that looks very similar to wild type, but yet is still different. So
Ron, if you take the spores from all the envelopes and plate them out, do you get these 4 different colony morphologies?
They did get different colony morphologies from most of them.
I see.
But they didn't get it in the environments where there was environmental contamination. There they were getting sort of overwhelmed with one type that didn't show that variation.
Mm-hmm. So that's actually a question I had. You can get B. anthracis from the environment? You can get it from soil?
You can, although only in certain areas, like in Texas, where there have been a number of cattle at points.
I see.
You don't normally find this if you go into an office building in New York.
But how about if I went into Central Park? Would I find it there?
No.
No?
No, I mean, no, because the interesting thing is that Bacillus anthracis it's associated with livestock, and I think primarily with sheep and perhaps cattle. So it's typically found, the spores are found in soil where there's livestock.
I see.
And so it's not, and also I think, well, now I'm confusing it, but it's not something you would expect to see in an urban environment, in the fact that, these people were coming down with inhalation anthrax, which is, you have to have massive amounts of, ingesting or, and taking in massive amounts of the spores to get that to happen. Because typically, Bacillus anthracis from the soil is the cutaneous form.
Right.
It's interesting to go back to the history of this particular strain,
Yes.
the Ames strain. It actually was isolated from a dead cow in Texas some years ago, and a culture was sent to USAMRIID. Now, the laboratory that sent it to the Frederick laboratory used a mailing tube. There was an old used mailing tube that had come from Ames, Iowa. And so when it arrived at USAMRIID, someone logged it in looking at the mailing tube and said, "This is from Ames." It was not. It came from Texas. It was never in Iowa.
It's great.
But, when the outbreak occurred in 2001, we had a great suspicion that it must have come from the USDA lab in Ames, Iowa. And in fact, the military sort of was responding to that threat from Iowa when the strain has never been there.
It's funny.
But that's very common because pathogens, like I used to work on Legionella pneumophila, and back in the early days, they were isolated from, the names were from where they were isolated. And there were lots of them that were strains that were isolated in England, for example. And I was working with Nottingham, and I'm going like, where in the United States is there Nottingham? And then I realized that a lot of the isolates came from Great Britain, so outbreaks from there. So it's kind of interesting.
So in this story, they had these 4 colony variants that they had isolated from the original envelopes of spores. And so they sequenced the total genomes of each of those 4, which is another remarkable feat in my view. And they found differences comparing the variants to the parental strain. And each of the colony variants had a unique change. What's amazing to me is that one of them had a single base change in this gene, which I think Cliff mentioned is involved in sporulation. A single base change leading to morphotype B, which is a larger kind of colony and different. Some of the other changes were more substantial. One of them was actually a duplication of 2,000 bases, which they had originally missed because the computers thought it was a mistake and they threw it out. I just love this because I never do any of this bioinformatics, but can you imagine that you would have missed it if you had let the computer crunch all of the data on its own?
Well, and it's so interesting that we would have missed it probably 20 years ago before sequencing.
Yeah.
We would have missed the whole phenomenon because, when Cliff and I were trained, the first thing you were told to look at is colony morphology, and colonies that looked as different as the ones in this paper, I would have guessed were different species, not even just different strains.
Huh.
And, you wonder how many strains were typed as different things based on superficial characteristics, just because humans are so visual.
Sure,
we count on our eyes more than just about anything else for an initial assessment. So I think this is so dramatically different from an outcome that would have occurred before sequencing. It's a really great example of how the technology has just completely turned around the field.
Sure.
I'd like to ask you, where would we be today? I mean, my sense is we would not be going back to colonies and picking out different morphotypes and doing whole sequencing, but we would be doing either metagenomic or we would be looking at single cell sequencing. Am I off in that, or is that where this sort of investigation would go if it was starting today?
No, I think the same approach as this paper presents would be taken because they had pure cultures of these strains, and I think typing them and looking for single base changes would be the same priority.
I think they said somewhere in this paper that when the investigation began, this technology, the sequencing technology, wasn't yet available, and they had to. This is something that just developed over the years. Not only the sequencing, but the software to be able to analyze it.
Yeah, when this first happened, it was a feat to have an entire genome of one organism.
Yeah.
And in fact, I think it's around this time that the first Anthracis and other genomes in that Bacillus group were completed. And the idea of just going in and sequencing 5 strains easily was really not on our horizon. Yeah. So it's kind of dramatic as well, just in the few years since then, how it's changed.
In fact, they say now that this should be a routine part of forensics, sequencing entire genomes. It's just amazing. So we have these 4 colony morphotypes, and so they say that these were found in the cultures from Bruce Ivins' lab. Ron, is that fair conclusion?
That's what is the FBI conclusion.
All right. So then I had a quote from one of the authors of this paper, Ravel. He said, "The science was one technique used to generate leads as part of a larger FBI investigation. Science tells us the spore came from that particular flask, but it's important to note that the science never pointed to Bruce Ivins. It was police work that did that." So, Ron, why is he saying that?
Well, first of all, because the spores could have been taken out in mass and cultured somewhere else. In fact, they were cultured after having come from the flask. The other conclusion was that someone didn't just scoop out some spores and put them in the envelopes, that they had been, in fact, grown beyond that. You don't know who did that. You don't know when that necessarily occurred. That means it's not just one individual that you can say definitively was the perpetrator.
So based on just this sequence, you could not say who had done it, right?
No, you can point to the origins of where the spores came from.
Right. But I think they said that these morphotypes are also found elsewhere besides in his laboratory.
I don't know that they have found another source for these 4 morphotypes, at least not the combination. But you can't preclude that they do exist somewhere else, having originated there.
Right.
Especially since some of them arise from single base changes.
Right.
Even though they weren't able to find them in the lab, it doesn't seem like it would be all that hard to find a single base change variant.
See, I wanted to go back to what Joe was talking about, being the dinosaur of microbiology that I am, is that, when you look at, I would have looked, if I was doing this back in the day, I would have noted that there were different colonial morphological types. And if I repeatedly had seen them, I would have looked at why they were there and just concluded that they were colony variants. And if they didn't have any bearing on what I was trying to do, I would just, note it in my head. But having worked on a lot of lipopolysaccharide stuff as a graduate student when Joe and I knew each other. Actually, Joe was the person who helped me get my PhD because she turned me on to the lab that was doing gas liquid chromatography of polysaccharides that enabled me to sequence my mutants. But so I was very aware of colony morphology changes and always used to look at the plates. But then when I worked on Neisseria gonorrhoeae, there are 4 to 5 distinct variants and colonial types. So I guess it depends on, your perspective of what you were looking at when you were studying bacteria. But as an old-time microbiologist, I always look at colonial morphology because I think I was told that is really something that, is really important to take note of. So I just like to think that, we've come a very long way, but the basic principles of microbiology still apply, and you can't just do bioinformatics and sequencing to address all issues, going back to, well, why would a single base pair change in a global regulator of sporulation result in a colonial morphology change that's so distinct? So, and that's why I like Joe's paper, because she uses, the paper we're going to talk about next, she uses traditional bacterial genetics as well as modern molecular biology to answer questions as well.
Yeah, so getting back to what I asked Ron, the last sentence of the paper, it says, using these assays, the investigative team was able to demonstrate that cultures within the FBI repository contained the 4 morphological variants together, which supported the FBI investigated efforts to identify the source of the letter spores. So yes, as you said, they could have been prepared elsewhere, but so basically the case doesn't lie just on this scientific analysis. There's a whole nother forensic analysis that the FBI ran.
Well, I don't know if the rest was a forensic, but it was a typical criminal investigation of looking at individuals, psychological profiles, behaviors, access, etc. The actual National Academy report suggests, though, that the FBI may be reaching a little bit too far in some of the conclusions based on just the science, and does, highlight that it's other things that you have to consider in a criminal investigation. And that, I think, really is an issue for microbial forensics. How far can it go? When we do a public health outbreak investigation, it takes a long time, but we get pretty close to what we think is the source. In this case, we get to the source, but not necessarily really what happened to it, who had access to it.
Sure. It just reminds me in a way of I don't know if any of you were aware of this, there was an HIV case very recently where two individuals infected intentionally their sexual partners and hundreds of sexual partners, and they could actually identify the origin of the virus in the partners and trace its evolution and say that this probably came from these two individuals. So in some cases you can, especially if you have a lot of variability and you can do a phylogenetic analysis.
But in that case, Vincent, the organism is still in the person. So the logical conclusion is if you start with somebody who's got one strain and it winds up in another person, and you know that direct contact, or at least mixing of infected blood, there's a better assumption. But taking a flask, from a flask of bacteria in a lab,
Sure.
You know what I mean? So to me, that's a distinction. And going back to what Ron said, in terms of public health or infectious disease outbreaks, it's pretty straightforward as to where the source was likely to originate from. But in those cases, we're not looking at it criminally. We're just trying to identify the source so we could stop the outbreak.
Yeah. Yeah, they also say here in the discussion, if a similar attack had been perpetrated with E. coli, salmonella, or any non-spore-forming bacteria, it would have been more difficult to attribute minor genetic variations to a source sample, which is something that we've talked about because there's much more variation if you don't form a spore. So I encourage everyone to read the FBI report if you're interested in the other evidence, which Ron mentioned, the psychological profiling and so forth, which they have used. So, Ron, why is this not considered a cut and dried case? The science alone can't prove it? Is that the problem?
I think that is the problem. I think that the determination that it must have come from this flask, the National Academy has questioned. It certainly points to this flask as the source, as the original source, but that it was taken from there, grown, and put into envelopes, the Academy did not think was 100% certain.
Okay. Anything else we should consider before we move on from this?
I just wanted to ask Ron a question.
Sure.
So does that mean that the fact that Ivins is no longer with us and there have been no other outbreaks, so they consider the case pretty much closed? They sort of got identified the source, and therefore there's really not much more to worry about in the future?
The FBI has certainly closed the case. They declared it over. There are members of Congress who want to continue revisiting the case. They do not consider the case to be final. So from the Justice Department, it's over. From the Congress, it may not be.
What about the scientific community?
I think there is a real division in the scientific community. Many of my colleagues who knew Bruce Ivins continue to insist that he could not have been the perpetrator. They just don't see him carrying out this sort of horrific attack. Others look at the scientific evidence and go, good enough for me. So I do get different reactions from my colleagues when I speak to them.
So you think this will be reopened at some point?
I don't. I think the Academy doesn't want to see it again. It was an extraordinarily difficult exercise for the Academy to go through. The FBI feels that they solved the case, and the Congress is so busy doing other things. I just don't see this getting reopened.
Certainly was a long and costly investigation in terms of not just money, but lives as well. So, but Cliff, you said, is it over? But someone could always come and do the spore release again, right?
Not, again, Ron would be able to speak to this better than me, but this was not you can't just go to Texas again and find a cow carcass and isolate, you know what I mean? It's like, these spores, at least from what I remember, were fairly sophisticated and weaponized.
No,
there wasn't.
Cliff, in fact, this report makes clear that all those reports that this was sophisticated, weaponized, were incorrect. But that certainly still persists out there in many of the news media reports. There was some silicon in the spores, but it does not seem to have been added. No one knows, reading the report, where that silicon came from. But there was apparently no weaponization. Someone simply took spores, presumably from this particular flask at USAMRIID, grew up a batch, physically dried it in some way, put it in envelopes, and sent it out as a lethal mix.
Well, because I guess I feel remiss as a microbiologist that I never, knew that. I mean, obviously, I even thought that. So it's good
and that's out there. Don't feel remiss. This is something that was like one line in this paper that said, by the way, they weren't weaponized.
Okay.
Thanks for that, guys. And our second paper also happens to be Bacillus. I didn't actually intentionally pick a second Bacillus paper, but I was looking through Joe's publications, and I found a recent paper, a quadruple enterotoxin-deficient mutant of Bacillus thurigiensis remains insecticidal. And I thought this was great. Could give us a nice discussion. And of course, I use BT all the time on my lawn. So, Joe, BT is a product that you use to kill insects in agricultural settings, right?
Yeah, that's right. BT is a bacterium that produces a protein toxin that crystallizes inside the bacterial cell, and then when insects eat those bacteria, then they die because the toxin is highly insecticidal.
So a broad range of insects can be controlled with this?
One of the nice things about BT is that it's fairly family specific, and so there are different variants for different groups of insects. So the one that kills caterpillars won't necessarily kill beetles or something like that. So they don't have as broad activity as many of our insecticides, the chemical insecticides.
We were for one of my other podcasts, we were in Florida, and we visited with the mosquito control division in the Florida Keys, and they use BTI to kill mosquitoes, which I guess is another strain which is specific for mosquitoes.
That's right. And the work that we did was based on Bacillus thurigiensis kerstachii, which affects the Lepidoptera, so moths and butterflies.
So I guess the origin of this work described in this paper is that there's a potential that toxins produced by BT might be ingested by people.
Yeah, it's been a very interesting debate that has been fascinating to me to watch evolve. I became interested in this work because my lab, when I first started as a professor, screened a large group of bacteria from soil and isolated from plant roots for the ability to suppress a very damaging disease of plants caused by an oomycete. It's a protist that invades and infects the roots of plants. And so we found that if we coated seeds with this strain, and it turned out to be a Bacillus cereus, we coated the seeds with Bacillus cereus. They would then germinate, and the plant roots would be colonized by Bacillus cereus, and there would be less disease. So we were interested in this strain as developing this strain as a pathogen control strategy. But what became very clear is that Bacillus anthracis, the one we were just talking about that causes anthrax, Bacillus thuringiensis, which is the insecticidal one, and Bacillus cereus, which we found as the biocontrol strain for plant disease, are very, very closely related. And in fact, it wasn't until genomic sequencing that there was a real indication that they had different evolutionary origins, because most of the activities that distinguish them, the insecticidal, the anti-protist, and the anthrax toxin, are encoded on plasmids. And so it seemed like the genome was pretty similar, and it is. Those three species really are not truly separate species because they differ in only a small number of genes. So when we started going to register the BC, Bacillus cereus strain, we thought there should be no problem because Bacillus thuringiensis had been on the market for almost 50 years. They're closely related. If BT is safe, BC should be as well. Yeah, exactly. And
it's funny that you're saying that because the Food Research Institute was right down the block from the plant pathology building. And
exactly.
In fact, Charlie Duncan, who used to work on perfringens, was doing work on B. cereus because it started happening in the 70s that there were B. cereus outbreaks. So I'm just laughing because, it's like looking at it from a different perspective. So sorry, Joe.
And, well, it's funny you say that because it was exactly those people at the Food Research Institute who first asked a question, well, aren't you worried about contamination of food? So we started reading about it, and what evolved over the years was very clear evidence that Bacillus thuringiensis and Bacillus cereus both produce these toxins that cause food poisoning and have either emetic or diarrhea effects on people. And there had been no problem, as far as we knew, we as a field, with Bacillus thuringiensis being used on crops very, very broadly, even though it had the same potential as Bacillus cereus. But it was interesting because the EPA, which was the unit that would have registered Bacillus cereus as for agriculture, said we can't possibly register anything named Bacillus cereus because that's associated with food poisoning. And so I said, well, if we put in the gene for the insecticidal toxin in those days, that would have been called immediately a BT strain. We could call it BT, and then would you register it? And they sheepishly said, yeah, probably. Well, we didn't do that because we thought that was a little underhanded. And we decided on the other approach, which was to delete the genes for the human toxins. And so we did that. First, we started in Bacillus cereus, and then it became clear that the EPA began to develop an interest in whether Bacillus thuringiensis was a problem as well for food poisoning because it had the same genes. So we went in and deleted the genes for the enterotoxins in BT as well.
So that's sort of a preemptive move because there isn't evidence that BT has been a problem for people, right?
Well, it's an interesting dilemma, and it really goes to the heart of sort of forensic microbiology, of being able to trace where bacteria came from. So, and also the way we diagnose disease. So when there's a food poisoning outbreak, people do a series of tests, and they often will find, if it's a bacillus, that it's a Bacillus cereus. But as far as we know, people haven't gone in and asked whether those strains have insecticidal activity. So, in fact, Bacillus thuringiensis strains might be the cause. And there's some evidence from groups in Europe that, in fact, some of the strains off the shelf, which are thought to be Bacillus cereus enterotoxin producers and had been isolated from food, in fact, do have the insecticidal toxin. So that's, we don't know. And so nobody is suggesting banning the use of BT because of this. But it's not clear that none of the food isolates are not BT.
So, Joe, is there interest from a regulatory standpoint or from an industrial standpoint in pursuing the quadruple mutant that you have? I mean, have you gotten calls from Monsanto or other companies that want to pick this up?
Well, most of the BT in this country, in the world, is sold by Valent Biosciences, which actually we used their strain in this work, and they were the ones who first requested that we do the work when they heard we were deleting enterotoxin genes in Bacillus cereus. They said, well, could you do it in one of our BT strains? And so they are interested in developing it, and I believe that they are trying to make a product that is presumably safer. Now, that's not to say that the original BTs are not safe. You're right. It's preemptive. It's preemptive of something we don't know, whether it could happen or not. But it's better to prevent it than not.
So you're going to benefit financially from the ignorance of or the safety of the American population.
We don't know. It's possible, though.
But what I wanted to ask, Joe, was that I know this was a laboratory test, but the big question is that if you took big deletions out of all of the putative genes, the putative virulence or enterotoxin genes. So the question is, from a fitness standpoint, did you do any field testing with these strains, or you couldn't do that because you'd have to get clearance for engineered bacteria or something?
We didn't do that. I assume that the company who owns the original strain would be doing that now. Mm-hmm. But it's kind of interesting, that you ask it, because as far as we know, there's no role for the enterotoxin genes in growth, colonization, or killing of insects. But it's interesting that in this strain, there were 4 copies of these very large operons. And bacteria don't accumulate and then maintain 4 copies of something without it having a pretty important role. And all of the strains we looked at actually had multiple operons for enterotoxin production. So someplace in Bacillus life history, these genes must play a role.
And that's really interesting. If you say, look at the relatedness of the Anthrax. This is an organism, or at least a genus, where everything is, I don't know about subtleus, but at least these 3, where everything is highly conserved. And so your point of, quadruplicate copies, really would lead me to think that they better field test it before they start, scaling up and selling it.
Yeah,
I agree. You know what I mean?
Yeah.
So you knocked out the G's for 4 enterotoxins, and then you showed, of course, that those strains are still able to kill insects, which I guess is the key, right?
Right.
So they still produce the crystal protein in the spore.
Yes, and that protein has no sequence similarity or obvious structural similarities to the enterotoxins that affect people. So it wasn't a surprise to us, but perhaps it was a relief that the strain maintained its insecticidal activity.
Are there other enterotoxin genes in BT, or just these 4?
There are a couple of others that have been very loosely associated with food poisoning. Basically, they've been found in strains that have caused food poisoning, but usually they're ones that have shown up once or twice, and there's no evidence that they're actually responsible for food poisoning. Mm-hmm. And just about every strain that has caused food poisoning has one or more of the kinds of genes that we deleted.
I'm wondering, how would such a toxin end up in your food? Would it be that the produce has bacteria, or would the toxin insinuate into the plant?
It probably doesn't reach us through the toxin itself. It's probably through the bacteria. And like a lot of food poisoning, Bacillus cereus food poisoning is associated with mishandling of food. But it's actually a pretty common type of mishandling. And one of the common products that has Bacillus cereus in it is rice. Mm-hmm. And because when you boil rice, you kill most of the bacteria, but you don't kill the bacillus because the spores are heat resistant. When you just put the rice in a cooler environment, the spores will germinate and start to grow. And so often people will leave rice on a very low flame on the stove for many, many hours. And those are just ideal conditions for Bacillus cereus to multiply.
It was really interesting back in the day when I was hanging out with people at the Food Research Institute. It was always Bacillus cereus outbreaks were frequently associated with, I don't know whether this was true or not, Asian restaurants.
Yeah,
And,
for
you know,
exactly the rice
Right.
reason that they would
Right.
keep big pots of rice going for a long time.
A long time. So it was very common that the outbreaks did occur in Asian, back those days, Chinese restaurants. But so it is a very interesting organism from that standpoint.
Chinese
So
restaurants and daycare centers, which was a connection I never quite understood.
were they daycare centers in Asian communities?
No, no.
I was just joking.
So there is some other group that seems to somehow propagate this pathogen.
Right.
So why does the bacterium have these toxins?
Yeah, I think that's the big question that we're interested in, that if they have maintained so many copies of the genes, what do these genes do for them that have been selected for over the years? And the only thing that we can think of is that either there's an activity of these toxins that we haven't identified yet, that it offers some sort of benefit to the bacterium, or perhaps BT and BC get into animals, cause this kind of diarrhea, throwing up, all the symptoms that we associate with the food poisoning. And maybe that's a way that Bacillus distributes itself and propagates in the environment. And that's completely hypothetical. We have no evidence to support it. But that's the only thing we've come up with that makes sense.
Sounds good. Viruses that cause gastroenteritis, the same thing. You have explosive vomiting and diarrhea, and it's a very effective way of spreading the virus to others.
But in the case of the Bacillus species, they're sporeformers, so theoretically they just need to be transiently in a host, and then they'll cause disease, and then either there'll be diarrhea or vomitus that would come out, and then they'd be things would dry up and they would form spores, and then some cow or something would come along and ingest it and not get really. So yeah, it makes sense that could be but being a spore former means that they're not obligate intracellular or parasites per se, so they can exist in nature, just a convenient way to amplify the population from time to time if you find yourself in the right place at the right time.
I've always found it interesting that Bacillus cereus, thurigiensis, and Anthrax have all accumulated toxins of some sort, and they're very different. They're not genetically related. The enterotoxins that are shared between Bacillus cereus and thurigiensis are, of course, but the insecticidal toxin is different. The antibiotic that Bacillus cereus produces that seems to be associated with plant disease suppression is completely unrelated. And then, of course, the anthrax toxins are a whole other class. So why? What do all of these toxins do for the bacterial life?
So let me understand, if I ingest some Bacillus cereus, how is the toxin elaborated? Does the bacterium multiply and produce toxin, and that is how I have poisoning, or is there some other?
No, that's why it's really called food toxication or food poisoning, and not food-borne pathogens, because it's not really a pathogen. The toxin has to be preformed in the food, because as far as we can tell, Bacillus cereus and thurigiensis don't grow in the human body. They are probably inhibited by stomach acid and just pass through the body. But each cell will contain a good dose of the enterotoxin, and that will be delivered from the food into the person, and the enterotoxin can survive to the intestines, and that's where it causes the problems.
So how does that get out of the bacterium into our intestine, the toxins?
Well, they have a secretion mechanism that enables them to secrete the protein toxins in the food itself.
I see.
So the toxins are probably all there along with the bacteria before we even ingest the food.
So the interesting thing is that it requires, when there are outbreaks, you have to find large numbers of bacillus in the infecting food, and as Joe rightly pointed out, you have to have high numbers to produce sufficient amounts of toxins.
Right.
So other organisms like Staph aureus, they produce toxins that are, again, staphylococcal food poison, I think, is the number one cause of food poisoning in the United States. And again, it's an ingestion of a toxin. Now, this is contrasted with the enteropathogenic E. coli that cause problems with undercooked meat. They're actually bacteria that are ingested they grow in the intestine and cause destruction to the epithelial cells and disrupt a lot of physiology of the cells. And Shigella, Salmonella, those organisms are enteroinvasive organisms. The gram-positives largely, like the Clostridium perfringens, Clostridium botulinum, Bacillus cereus, all of those guys produce toxins that are ingested. Mm-hmm. They don't actually live inside of the intestines of human beings.
Okay. I have one last question, and that is, when you use BT as an insecticide, do you ever select for resistant insects?
There's surprisingly little resistance from the use of Bacillus thuringiensis as an organism, and that's after many, many years of it being on the market for organic farming and gardening and other uses, because it's one of the few ways of controlling insects that's approved in organic production. But now the Bt toxin gene has been taken out of Bacillus thuringiensis and put into plants. And so there is a large number of acres in this country of plants that produce their own Bt toxin, and they're thereby protected from insect damage. And so those are called genetically engineered plants, and so that's the whole controversy over GMOs has focused on, among others, the Bt-containing plants. And there's been a lot of concern that we would lose the ability to use Bt in any form, either the genetically engineered form or as the bacterium, because there's so much selection pressure with all of these millions of acres of crops producing Bt toxin. So the companies that produce these Bt plants have gone to great lengths to ensure that farmers plant what they call refugia, where insects that are sensitive to the toxin can thrive. And so those are just plants without Bt, so that we always maintain a mixed population of insects that are resistant and sensitive to Bt toxin. And so far, we haven't lost the use of it, although there had been observations of resistance out in the field.
Okay, that's great. So that's a paper in Microbiology. The authors are Klimowitz, Benson, and Handelsman. And I thought that would be fun to talk about. The last thing we have on our agenda for today is a few emails, and that's something that we have been getting increasingly at TWiM. And let me read a few of these. The first one is from Hubert, who writes, I'm a Belgian student in veterinary medicine. I hope my English isn't too bad, and I'm interested in microbiology. I have a few questions about the last episode of TWiM, quite a lot about episodes of TWiV, but I'm still in the process of listening to all of the episodes. The first questions are about the Yersinia pestis infection story. During the explanations about the life cycle of Yersinia, you talked about iron as the main limiting reagent in many infectious processes. Is it only because of the fact that it's not easily available in the host, or is iron a more important reagent than other trace elements like zinc or manganese? Also, are the requirements for such reagents the same in bacteria as in eukaryotic cells? I suppose not, since there are already some variations among domestic animals, but in that case, are the differences important? Who can tackle that one?
I could attempt to tackle it, Vincent.
Sure, go right ahead.
So iron is a very important trace element, or a very important mineral for most life, and most microbial, or at least bacterial species have developed very elegant ways to acquire the iron, because it's a necessary element required for the growth of most organisms. So it's as far as manganese and magnesium, they're not as vital as far as I know. I'm not a biochemist, and I forgot all my biochemistry stuff, microbial biochemistry and physiology, but iron is clearly very important, and perhaps more important, at least it needs to be available to bacteria to grow in hosts, as far as I know, and in the laboratory for that matter.
It's also less available because of the insolubility of some of iron's valence states. And so, for example, in soil, iron is often limiting, and manganese and zinc are much less often in limited quantities.
Of course, in eukaryotic cells, well, at least in people, we need iron for being part of hemoglobin.
Right.
Zinc, yes, zinc proteins all require zinc, magnesium for sure. You need that for nucleic acid enzymes, and manganese to a lesser extent, I believe.
Don't cytochromes also have iron?
Yes.
Some do, yeah.
Yeah. So in bacteria, bacteria that undergo aerobic respiration need it when they're using the TCA cycle. So unless you're completely anaerobic, it's probably not something you could do without.
The other question is about the microbiome. You talked about the NIH microbiome project and studies about bacterial microbiome. Is there a similar project about a viral microbiome going on? I don't know if it's at the project level as the NIH microbiome project is, but certainly there have been a number of studies. Of course, the ocean virome has been sequenced. The gut virome has been sequenced a few times, but there are certainly more places to look, and I think that's a growing area of virology for the future. Finally, could you make an episode about bacteriophages and their relations with bacteria and phagotherapy? Sure.
I'd love to do one on that.
We could do that. Thanks a lot for all your podcasts and blogs. It's really informative and feeds my passion for microbes more and more. P.S. I plan to do a PhD in microbiology after I get my diploma, but I hesitate between bacteriology and virology. Both are so fascinating. How did you choose your area of research, and do you have advice to choose between one or the other? I'd be interested in what you think about that. Joe, what do you think?
Well, I'm partial to bacteria, but that's not because they're more important or intrinsically more interesting. They're just more interesting to me than viruses are. I think broader PhD programs are probably better than more narrow ones, so I would always recommend that somebody get a degree in microbiology over virology, but whether they choose to actually do research in one or the other, I think is a personal and almost aesthetic decision. That's how most of us choose our fields of science. We find one just more attractive than others.
How about you, Ron? How did you choose bacteria?
Well, as I said, Selman Waksman pushed me away from the viruses. But, in answer to the question, I think you go with what you really like and in the environment in which you get to work. Having great mentors is just so critical to advancing your own interests and career. So find a place with lots of people in diverse areas of microbiology, gain as much as you can, and then pick the area of specialization you fall in love with.
How about you, Cliff?
Well, I went to undergraduate school with you, Vincent, and I knew you were more interested in viruses than bacteria, so I figured I could never compete with you, so I decided to go the bacterial way. Actually, I was always fascinated with bacteria, particularly those that cause diseases, and I just thought it was really cool to be a single cell and be able to live almost anywhere that I could think of on the planet. So, and just to echo what Joe said, I got a PhD in bacteriology at Wisconsin. I think Joe did too.
Nope.
But no, you didn't?
No, I got a degree in molecular biology.
Sorry. I didn't mean to denigrate your degree, but
oh no! I wish I had a bacteriology degree.
But the one thing that I've learned is that by getting the degree in bacteriology, which includes microbiology, I just feel like I understand viruses well enough, and I understand nitrogen-fixing bacteria, and I understand bacteria that cause disease. And by studying microbiology, it all makes sense to me that it all fits together. So like Joe said, I would train in microbiology, but then just have that background, and then if you like viruses better than bacteria or vice versa, go with what moves you from a research perspective.
Well, if I could add my thoughts, I read a book which got me excited about viruses when I was in college. I read Fever, which was the story of the discovery of Lassa fever virus, and so I thought that was great. I had also read Paul de Crieff's Microbe Hunters, and I thought it was great, but the virus thing really excited me. And as Ron said, I found a good mentor. In fact, two great mentors, and that was the key. So I think you have to just decide what you like and then find people that will teach it to you really well, and then it will work. So in the interest of time, we'll save the rest of the emails until next episode. And I just want to tell everyone that you can find TWiM on iTunes at the Zune Marketplace or at microbeworld.org/TWiM. If you like TWiM, tell others about it. Links and reviews in iTunes are particularly helpful. And as always, we'd love to get your questions and comments. Send them to twim@twiv.tv or go on to microbeworld.org/twim and leave a comment there. If you have a story you'd like us to talk about on TWiM, put it on microbeworld.org and tag it with TWiM. And I'd love to thank everyone for joining us. Joe Handelsman from Yale University, thanks for coming on.
Well, thanks. And don't forget to thank your microbes this week.
Microbes, we need them.
Yes.
And we look forward to having you back. Ron Atlas is at the University of Louisville. Thanks for joining us.
Great being here.
Hope to see you again. And Cliff Mintz is at biojoblog.com. Thank you, Cliff.
You're welcome, Vincent.
We'll see you next time. I'm Vincent Racaniello, and I'm at virology.ws. I'd like to thank ASM for their support of TWiM, Communications Director Barbara Hyde, and Chris Kondayan and Ray Ortega for organizational and technical help. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.